let's welcome Professor Frangi for the Insight talk.
Thanks a lot, Hao, and good afternoon to everyone. So it's a pleasure and honor to be able to talk to you about some of the work that we are doing in Leeds, in basically at the interface of imaging and modeling. And the work that I'm going to present is largely funded by the Royal Academy of Engineering. So I have a 10-year award for actually focusing on this area of computational medicine and regulatory science. So the talk has two parts. One part where I'm going to try to explain a bit what is the story behind everything what I'm doing and a bit the vision, and then a second part where I'm going to explain some of the progress we have made in the direction of this vision, so a bit the action behind. And I would say that the summary of all the work that we do in our group is to work on developing models of both organs and disease and therapies from individual images, particularly from large sets of individual images, and then be able to use those models to guide personalized medical device design or personalized medicine, personalized interventions, interventional planning. And this is work that we've been doing with a number of colleagues over the years. So basically, when you try to improve a healthcare system, what a number of people have identified in the literature is that there is both the importance to improve access to the clinical knowledge that we have about that particular disease, but also about the patient information that we have access to. And the way I tend to think about this is as a sat-nav, where in order to be able to do personalized guidance to a particular outcomes, we need both to understand the GPS positions, and we need to understand the patient's specific positions. We also need to be able to create maps that describe the territory, the disease that we need to navigate. And you could imagine, what would be possible with a sat-nav if you have either a very faulty sat-nav that doesn't give you the position well, or if you have a very good position but you don't have a map. Both of things will be, unhelpful. So that implies that you need to be able to do analysis and modeling, not only at the individual level, but also at the populational level, so that you can actually derive those maps. So some of the questions that I'm trying to address through computational medicine, which is basically the use of engineering, math, and computer science to improve both our understanding of disease, but also of the possible treatment strategies. So some of the questions that I address there is basically, what constitutes normality or disease in terms of patterns in a population, how you identify patients that are at risk within a population, which patterns of, which patients will benefit more from a particular type of therapy, and so on. And some of these questions will emerge today. And some of the ideas that I will present today are described in the editorials that we wrote over the last, a few years in media and in TMI that you might be interested in having a look at. Now, the whole idea behind this approach is that we are inundated by data, and we see that more and more, but it's not enough. I mean, what we really need to have is wisdom, which is essentially validated knowledge, which is in itself structured information, which is well processed and quantifies data. So that based on this validated knowledge or wisdom, we can actually take decisions about how things may evolve in the future. So as long as the only thing we have is data, even if it's, quantified data, the only thing we know is something about the present or the past of the patient, but ultimately clinicians want to take decision in terms of different course of actions. And in order to do that, there are different ways to traverse this pyramid of abstractions. And it's either by using phenomenological algorithms or methods or AI, which essentially you use when there is abundance of data, but little knowledge or principles, or what we call more mechanistic AI, or techniques that you use when there is very clear first principles from maybe physics or physiology or biochemistry or anatomy, but actually that mechanisms are not, we need to instantiate those mechanistic principles into a particular individual, so into the data of one particular subject. Now, this is quite important because if we look at predictions that some people have done about how the demands on computing and storage will evolve over the next few years, So this is a model developed by Leo Helbing in ETH in Zurich. And what he shows is that while the computational power, as we know, grows exponentially and doubling every 18 months, like the Moore's law indicates, data volume is currently doubling every 12 months, also being exponential. So there is an inflection point where there will be more data than processing power, but also that this data volume is accelerating. It's now doubling every 12 months, but people talk about it doubling every 12 hours at some point. And more importantly, there is a systemic complexity, both in the algorithms that we developed that have more and more parameters, but also on the complexity of the disease, as we understand that with increasing number of comorbidities and interactions between the disease, that actually give grounds to believe that this complexity is going in a factorial way, which is much, much faster than exponential growth, which means that we will be very soon in a situation, if not already, where the complexity is so big that there is no amount of data that we collect that can be helpful to identify the parameters of those systems, and there is definitely no processing power able to actually amass that data in the first place. So that's a very bleak perspective in a way, but what I think comes into rescue is that in order to overcome this problem, you need to incorporate priors. And that's where techniques like physics-informed machine learning or domain knowledge-informed machine learning becomes helpful, because what essentially these approaches say to us is that there is no need to estimate all of those parameters, because we understand how some of them behave based on first principles. So just as an example, techniques like physics-informed or model-informed machine learning are actually quite important. They incorporate physics or knowledge in the form of differential equations, for instance, in the loss functions, and that allows to actually reduce the error bounds in our predictions, as some colleagues have articulated in a very elegant way. Okay, so how does this all relate to medical devices now? Well, on the other side, if we look at this on the application side, there is a big problem out there, which is that if you look at the last 10 years, as some investigative journalists have done, there is a huge amount of death that have been produced by misfunctioning of devices that went through regulatory approval, FDA, and other regulatory approval processes, while there has been nearly 2 million in the US that have serious adverse complications. So this is really, really bad if you think that all of these devices, are from all different positions within the body. So it's not like one particular device has all the problems. And second, that all of these devices went through regulatory approval. And this will happen more and more, because as technology becomes more complex and new devices and new technologies start bringing together different technologies like digital technologies with new materials, with new biochemistry, for instance, the complexity of the devices will mean that it's inevitable that you need to think about novel ways to regulate and evaluate them. And when this fails, there is all kind of other things that start failing that I will not go in detail because of lack of time, but ultimately, there is a longer time to market and R&D costs, and also a lower trust in the industrial sector and shareholder value, which is all bad. So what we are trying to develop with some of the things that I will show later is ways in which we can reduce the amount of testing that has to be undergone by a device from years to months, while at the same time maintaining, if not improving, our ability to ensure safety and efficacy of the devices we test. And that has a number of advantages in terms of if you could do through computational modeling evaluation and testing, that means that we could fail faster, earlier. We reduce the risk to patients because we only do trials that are, with devices that are likely to be successful. We get better outcomes to patients because devices are better tested, and a number of other improvements, including reduce harm to animals, because we need to do less animal testing as well. So how do we do nowadays testing, and what do we mean when we say regulatory evidence? Well, regulatory process require the provision of scientific evidence. Scientific evidence on safety, so this device is unlikely to cause harm, and efficacy. This device actually makes a difference and improves life. And currently the three ways to do that is by using animal testing, bench testing, and human testing. And this happens at different stages of the device process. And by the way, I'm talking about devices primarily because that's where I work, but similar concepts apply for drugs, although I'm not working in that space myself. But there are other people who are working in that area. So what we advocate is that there is a fourth route for testing, which is called, which we call in silico trials or computational modeling and simulation, which is the idea of replicating in digital twins or virtual replicas experiments that we would otherwise have to do on humans. The advantage is that if you can do a replica, you don't need to worry about replicating, bench or human or animal testing, because usually these are surrogate to come to humans that you do when when you're in very early phases. So we can shortcut some of the needs for animal and bench testing by doing proper in silico testing. Now, this is in a way not particularly new when you think about the way industry works in general. So if you look at the automotive industry over the last 20 years, there has been massive improvements in the products, but also in the processes that we use to do them. We don't do aerodynamic wind tunnels where you put, sort of winds, simulated winds and dies to look at aerodynamics. You actually do computational aerodynamics, and you can do virtual prototyping and AI self-driving cars. So the technologies of modeling and simulation, virtual prototyping, and AI are actually used more and more in other sectors, but surprisingly not so much into the medical domain. So if you look at 20 years worth of development in the context of a particular device, in this case, endovascular embolization devices for aneurysms, so coils, stents, and flow diversion devices, you see that over 20 years is fundamentally still a bunch of wires. And the worst part is that you still have 20% nearly complication rates and nearly 3% mortality rates. And one wonders, why couldn't we do the same things we do in the automotive industry by using, modeling and simulation on anatomical models that are derived actually from medical imaging? And that's where this connects with the work that many of us do here. So the idea basically is that we can go from a situation where we do very little computational modeling, lots of bench, animal, and human testing, to a situation where we do more virtual patients, more computational modeling, so that we still may need bench testing, animal testing, and humans, but in lower proportions, because we only do that with devices. So we only do human testing with devices that have passed the test of computational evaluation. So the hope is that there will be less devices. There will be more devices that will fail at the computational phase, and therefore we will not have to do a human testing, and we can put all our efforts in human testing only in pivotal, essentially validation studies for devices that we have a high confidence that are going to be successful. And with respect to animal and bench testing, rather than being used as a preliminary step towards human testing, most likely going to be used as ways to validate the computational modeling themselves. So the idea is that we will be able to replace, partially at least, reduce, and refine the trials that we do in humans by better use of computational techniques. Okay, so just to give you an idea about how this looks like, imagine that we have libraries of anatomical models. So in this case, what you see there is a subset of a large library we have on brain aneurysms. And each of these digital replicas, what we can do is we can create a numerical simulation of the hemodynamics by putting virtually a stent in each of those. And what this means is that for each geometry of our library, we will do a virtual implantation and we will look at flow. And normally what we look as variable of success is what we call the flow reduction regime. So if you look at the flow within the aneurysm before you put a device, and you compare that to the flow after you put the device, you look at how much you have reduced the flow in the aneurysm, and that is what we call flow reduction rate, which is this percentage here. Okay, so in that case, for instance, this particular device is likely to have responded less than this other device to the presence of the stent. Now, we can do the same device at different anatomical configurations, like placing it in different real patients. But you can also look at what happens if I take the same device but I by now compare different physiological regimes like rest and exercise, and I can do that by having models, Gaussian process models in this case, of how the boundary conditions, so the inlets and outlets of flow in the aneurysm behave over a population of subjects. So what you see here is the flow rate for stress conditions and for rest conditions. And what you see in a shadow around the ball line is the variability, the uncertainty we have in the flow conditions over a population of 100 subjects. And then finally, of course, you can do the same thing, but now with different devices. So you have same geometry, same physiology, but now different devices themselves. Okay? And for each of those cases, we have an idea about how, for instance, the flow has acted, and the higher this number, the better it is. But what you can see is that this number can have big variations across different conditions, which is something that we observe, we have experienced in real trials, that actually devices don't always perform well and in different types of subjects. So what an in silico trial ultimately is a massive, ensemble simulation space where every node in here corresponds to a particular anatomy for a particular physiological condition for a particular device. And where this links with what we do in medical imaging is that producing these virtual experiments in a fully automated, streamlined manner requires advanced image analytics, among other things, available to us. The other thing is, where do we get this data? Well, we get this data from large repositories of real-world evidence or of clinical population data sets, like, for instance, UK Biobank. Okay, so I'm going to talk, most of the talk today will focus on brain aneurysms. We have two areas where I'm working at the moment. One is flow diversion devices, the other is TAVI devices, but this is a bit more work in progress. So I'm going to focus primarily on flow diversion devices. And just to give you an example about how serious this is, I want to give you the only example I'm going to give you on TAVI is about showing what happens when things go wrong. Okay, so TAVI, one of the larger imaging manufacturers in TAVI, Boston Scientific, developed a trial called Reprise 3. This trial collected about 1,200 patients, is about like 20 terabytes of data, just to give you an idea, this RCT, this randomized controlled trial. And this trial this trial cost about $80 million, took about 6 to 9 years to produce. And then in November 2020, this device called Lotus was recalled from market. This is all public information, so I'm not disclosing anything secret here. But this device was recalled in November 2022. The cost of the recall was between 200 and 300 million for the company in losses. Okay, so this is just one device, Lotus Edge. This is just a bigger sample of all the devices available in the market. So imagine for each of these devices, you need to run a trial like this, and you have potentially the risk of failing. This is massive in terms of the cost, okay? So that's the problem that we are trying to solve, is not failing at that later stage, because it's very expensive, very risky, and very, inefficient for the healthcare system. Okay, so in terms of going now in a bit more case study, the way I would like to do this, and I'd like to emphasize, this is work that happened in my lab over about 15 years. So there's a lot of work that I will present in a very, high level way. But the way to do that, I want to start by the end. I want to show you a publication that we did last year that for us was the climax of a long period of work. And then I want to dissect some of the algorithms that are behind that paper, so you understand the technology behind, okay? So one of the areas that I've been working now for nearly 20 years is hemorrhagic stroke and intracranial aneurysms. So these are bulges in the vessels of the brain. They like to happen mostly around the bifurcations of the vessels. And then there are different ways to treat them, but fundamentally there are three. Either you need to do craniotomy, so you need to open the skull and put a clip. And That, as you can imagine, is very traumatic. It takes about a month, the full recovery, and is very dangerous, but it's the way in which this treatment started and is very efficient in the sense that it removes the aneurysm fully from the circulation. Now, because this is very invasive, what a few years ago, well, quite a few years ago now, has been developed is a technique which are minimally invasive. You enter through a small incision in the groin and you go under image guidance all the way to the brain. And if you do that, you can either put coils like this, or you can put coils, but if the neck is very wide, you can put a scaffolding stent, or you can put a low porosity flow diversion device, so a low porosity stent that effectively is full of little holes but reduces the flow such. Now, why we are interested in doing these weird things about putting all these wires in there? Because ultimately what we want, what all three devices do, is to reduce the amount of flow within the aneurysm. If you do that, the aneurysm will clot, and as a consequence, you close it from the circulation, right? So you are trying to produce selective clotting. That's kind of the idea behind. Now, in order to develop an in silico trial of a device like this, what we need to do is, on the one side, find ways to build virtual patients and populations. We also need to be able to do virtual examinations and interventions, so how we insert a device, and then we need to have virtual outcomes or virtual readers, okay? And I'll try to explain a bit that in where all these components come. So I explained a bit, this is like a different picture of the same devices that I explained you before, so that you have a view about them. So this is the clip, this is the coils, this is a stent, which is used for coil assistance, and then this is the flow diversion devices that if you put these devices, you don't need the coils. To give you an idea, each of these devices can cost between 5,000 and 20,000 pounds, and this coils is about £1,000 each of them. So this explains a bit what I said earlier. We have an aneurysm, there is flow. We put a flow diversion devices, you reduce the flow that produce thrombosis, and as a consequence of the thrombosis, the aneurysm sort of is removed from the circulation. That's what we're trying to do. And this is the device that we're going to be talking a bit about, which is a Medtronic device, and it's called Pipeline, or PED, or Pipeline Endovascular Device, and it's one of the most used devices. Now, we're going to be focusing primarily on the device. You could do also in silico trials about the delivery system, so the system that you use to bring the device in the first place. If you remember the study that I was talking about on the Lotus device for TAVI, the main problem of that device wasn't the device itself, but was the delivery system. Okay, so the delivery system is very important as well, although we are not going to focus on it today. So this is the sort of evidence that a device like the PED uses to get regulatory approval. So this is the first publication, what is called the PUFS trial, or Pipeline for Uncoilable or Failed Aneurysms. This was published in Radiology, as far as I remember. It took about 8 years from design to publication, 6 years of recruitment. And this is to get in the whooping number of 108 patients. So it's not massive, okay? And probably this trial cost between 20 and 40 million. Now, the interesting thing is there is 3 of these trials. So this device, if you go to the webpage from Medtronic, they pride themselves to be the most studied flow diverter worldwide, with Premier being one of the trials, 141, PUFS 106, and Aspire 191. So if you put these 3 trials together that I'm going to talk about in a minute, they don't make 500 patients, okay? So that's all the evidence with which you go to the market. And to give you a sense, in our local hospital in Leeds, we do probably 100 patients a year. So if you multiply by the number of sites across the world, we are putting thousands of these devices a year. And you do that approved with 500. So we developed and published, and we are very proud about this. It's a paper we published in Nature Communications last year. We developed a trial that actually uses all the technology that I explained before. And what we try to do is predict, so we call FD-PASS to our trial, we try to predict the efficacy of the PED device. The FDPAS also explains, device failures scenarios, so when things go wrong, explains, how we can use these models to explain failure scenarios. And also we have some new hypotheses of situations where you shouldn't be using the device. So to give you an idea about how the pipeline looks like, and if you remember my triangle of going from data to wisdom, what we are trying here is to produce wisdom about the device based on data about the device through a complex processing and modeling pipeline. And that pipeline has components for segmenting images from rotational angiography, created, nice 3D models. They need to be really nice because we're going to do fluid dynamics on them. So they need to be watertight meshes with, high quality tetrahedral meshes. Then we need to have algorithms for deploying the devices themselves, models for modeling physiological regime. We need to solve the Navier-Stokes equations that ensue from this process. And then we have virtual images where we have physiological simulations. But it's not just flow. We also have a subsequent biochemistry model that provides, so these are streamlines for the flow, and here you see the streamlines as well. But these color code maps that you see here are not flow. They correspond to the density of and quality of the thrombus that is being formed. Okay. And this is this clotting model is a system of equations of ODEs that is coupled on top of the fluid dynamics. So if you are able to do these simulations with multiple subjects, so you have in the end vectors like this, where you have the original images, detailed vascular models, segmentations within the segmentation of the vascular to detect the aneurysm, detailed characterization of the aneurysm, hemodynamics, simulation results, and then all kind of additional metadata we know from the patient. So it's a complex vector from where you might want to do different type of things. So what we did in that trial is utilizing this pipeline to then do a retrospective selection of patients that satisfy the same inclusion and exclusion criteria as these 3 clinical trials that I want to compare against. So we ended up with a subpopulation of subjects that has the same demographics. And in this table, what we observe is the FDPAS, which is our study, and the 3 conventional trials that were done on this device, and where you see that we tested that the demographics of these 3 populations are pretty much indistinguishable to each other, and therefore we can use our population as a surrogate to the populations of those trials that we didn't have access to. One other important point is we actually had only about 82 aneurysms, but you will see here 164, because for each of them we could reproduce both normotensive and hypertensive conditions, which is interesting because in most of these trials they couldn't compensate or correct for hypertension, and you will see that this is actually quite important. So if you run this pipeline and you then look at the efficacy of these devices, which is measured by the ability to have angiographic occlusion of the device, and I could go in more details in the questions if you're interested in how we measure efficacy virtually, but trust me, there's a way to do it. And if you do that, what we show is that the overall efficacy as measured from this trial is pretty much bang on the numbers of the 3 trials that they did clinically. The difference, each of these studies took 6 to 8 years. We did that in 3 months, okay? But the other thing is, if you see a bit more in detail here, there is a shaded line, which is 83% for normotensive and 67% for hypertensive. So we could also look at what happens if all the patients are normotensive or all the patients are hypertensive. And what we see is that the response rate is massively affected by hypertension, which is something that was anecdotally sort of known, but there was no trial that actually made the point clear. So basically what we did is we could explain a number of failure scenarios. So these are failure scenarios that are known anecdotally in the literature, but no trial that explained that. And without me spending now a lot of time going through each of these plots, but fundamentally what we did is we could explain each of those scenarios based on simulations. And most importantly, we could analyze the effect of normal tension and hypertension in each of these different scenarios, looking at in which cases hypertension was an important factor or wasn't. The other thing that is quite interesting is that we could not only replicate what those studies do, but also have provide additional insights. So this aneurysm you see here is, if you look at this first row and the lower row, is the same anatomy, the same geometry, the same subject, but in the case 3 is normotensive conditions. So we control with the boundary conditions that the blood pressure was normal, was with a normal physiological rate. Well, this one is hypertensive. So this mimics the patient being hypertensive, but otherwise identical. And what we saw in this simulation, but this was consistent across all cases that have a branching aneurysm, a branching vessel. So this is the main vessel. You see the aneurysm here, and then there is a branching vessel on the side, is that all the aneurysms that are hypertensive and have a side branch, that are normotensive and have a side branch, they have a good patency in the side branch. However, if you have hypertension, the side branch becomes being clot as well and produce a stroke. Okay, and this is a complication that has been seen in the literature, but hadn't been fully explained before. So what are some of the techniques behind this? And I'm going to explain some of those steps. Well, you need one, the traditional one, you need segmentation. But it needs to be segmentation that is mindful that you might work in different modalities, that if you do a simple, isocontouring, even in rotational angiography, that is probably the simplest of all of these techniques, you will be missing vessels or, having segments that come in or out, right? And this is quite important when you look at hemodynamics. We also have situations with kissing vessels that touch each other because the resolution is, they're in the limit of the resolution, and you don't distinguish these two vessels as being two different running tubes parallel to each other, but you merge them as if there was a single one. Or you can have missing vessels that are lacking in between two different segments. And all of those will bring problems for when running numerical simulations. So developing AI algorithms that are able to run at scale and are able to segment small vessels and large vessels with high accuracy remains being a challenge. And we've done some work in this space. We used to have level set based algorithms that took quite a long time to actually run, and now we have techniques that actually run the whole pipeline in 30 minutes, which is actually quite good. And the whole pipeline is not just segmentation of the vessel, but is detection of the aneurysms, is center line, is detection of inlets and outlets, is being able to place the neck of the aneurysm, release the stent, generate, do meshes. So it's quite a lot of the steps, all of which are sort of computational geometry / computational imaging, and requires expertise from, like the one that many of you and myself have. Another important thing is, for running simulations, you want to segment ideally the whole circle of Willis. The problem is many of the clinicians, these data are not usually annotated, and if they're annotated, they only look in the area around the aneurysms. So we develop techniques that only with partial annotations you can extrapolate to the behavior outside the region of interest. And the idea is that with manual segmentations like this, we can then extrapolate and segment the rest of the vascular tree with high accuracy. I'm not going to go into the details of every figures and every number here, but what I want to emphasize is we have done a thorough validation of each step, because that's a fundamental point for having confidence that you can run these on a large pipeline and have results that are reproducible and are repeatable. The other thing is that many of you, if you are familiar a bit with trials, you will know that there is something which is called selection bias, and it's that when you are selected to be participant in a trial, usually there is already a lot of constraints. I mean, that means that you have the disease, it means that you walk through the hospital and you're able to go to do imaging. So all of those things make that when even if you run a trial with those devices, when you then go to the general population that has a lot of those covariates, let's say, don't hold anymore, then your performance goes down because you're on the wild, essentially, right? So one question is how we can develop virtual populations that don't replicate only the populations of trials, because we know their biases, but replicate rather the wider population as a whole. So then working on imaging, population imaging resources like UK Biobank is great, but then you have the problem with population imaging studies that most of them are based on volunteers, and therefore none of the imaging techniques are invasive or use contrast. So for instance, in UK Biobank, you have T1, T2, and proton density brain imaging, but you don't have any angiographic imaging. So we've been working in the problem of, can we actually, based on imaging that is like the one in population imaging studies, derive angiographic data like the one we would like to achieve? I cannot give you a lot of details about how the algorithm works because it's in the process of being filed a patent, but what I can show you is some of the results, and where maybe this is probably the most interesting one, where I'm showing here two synchronized maximum intensity projections, where one of these two images is a real image and the other one is a synthetic image. The other image has been synthesized to match the same intensity than the real image here, but with but is only based on T1, T2, and proton density. Okay, and essentially this is a Turing test where I'm asking you, you know, which one you think is the new one, sorry, the real one, or the real one. So this is another view. This is a maximum intensity slab projection. They're actually quite difficult to distinguish, but the synthetic one is the one on the right. Okay. So imagine now with these algorithms, we could actually run this algorithm on the full UK Biobank. We can have access to, you know, 40, 60,000 subjects, brain scans, you know, without any having to do any further data collection. And that means that we will have a very large population study from where to test different devices. And again, I'll save you all the details, but we've done a lot of validation in terms of anatomical detail of the vessels, but also in terms of hemodynamics. So like what happens to the hemodynamics if you run it on the real segmented vasculature the segmented vasculature on the real images versus if you segment the vasculature in the synthetic images. And so this is the ground truth and this is the synthetic, and what you observe is that the results are pretty much indistinguishable in a hemodynamics point of view as well. So just to start a bit wrapping up, what I like to show now is also that we developed techniques to do numerical flow simulation. Now this is validated quite a bit. So we looked at variability and sensitivity of these simulations to different boundary conditions. We know that the primary source of variability is the anatomy, so segmentation is very important, and then perhaps boundary conditions in the second step. And again, I don't have the time to go into a lot of detail today on this, but we've done exhaustive evaluation and comparison of these, techniques in the last, 10 years of our work. We also are able to do virtual stenting, and this is a lot of what I'm going to talk to in the next 2 or 3 slides is actually deformable models and simplex meshes. So again, lots of connections with our community, but where you can look at hemodynamics in different configurations and look at how the flow has changed depending on whether you put the same device in one direction, in the other, or you put both. Okay. And that means that we can do now with simplex meshes very simple device insertions that we can place into the particular aneurysms, and we have then tested again and validated against physical and numerical results. So for instance, we worked with colleagues about 10 years ago with colleagues in Milan, where structural mechanics, and we demonstrated that the results are within, reasonable tolerance. We also did comparisons between real and virtual angiograms. So what you see here on the left side is real angiograms, where you see the device here, and we put this, well, I hope you see the device. You can see it there. But these devices are usually not visible. You see that you only see the points at the beginning and at the end that mark that are marked for visibility, but usually they tend not to be visible within the body, so that you don't affect the visibility for the interventionalist. So we sort of outline it, the envelope of those, and we then compare it with the same anatomy with we did a 3D printing of the device, and we sort of inserted a real device. But then in the digital replica, we could do a virtual stenting. So we had the real and the virtual sort of side to side to the comparisons with two different stent brands. And then what you can do is inject contrast agent through the physical phantom. So you put that into a programmable pump, and you inject contrast agent, and you look at the swirling pattern and the time dilution curves, which is what you see here. But you can do also simulated virtual angiograms numerically, which essentially is solving the transport equations of a bolus of contrast agent. And then you have time dilution curves from the real and the simulated. So the green and the pink correspond to putting a sample volume in the middle of aneurysm and look at how the dye increases and decreases over time. The blue, by the way, which is very different, is a reference point here in the inlet that is used as control. But you can see that actually the numerical and simulated time dilution curves are pretty much identical in those cases. So you can then run simulations where you look at the aneurysms in no stenting condition and with different type of stents. And that's the basis for a lot of the virtual implantations that we did in the past. And we also have done some studies on patients where we insert in the devices and we look at clinical outcome from those. Very, very quickly, it's possible to do the same things with coils. Coils are, in a sense, a bit more complicated to model because they're more like an stochastic process. So what and there is tons of variabilities depending on memory, shape memory that they have, and so on. So what you do with these coils is you keep adding number of coils to make your density or your packing high. We talk about packing rate or packing density to the volume of metal over the volume of the aneurysm itself, and the higher the packing rate density, if you don't overpressurize the aneurysm, the better, because you make sure that the flow goes really down and therefore you coagulate. So if you look at these situations in a surgical specimen, you see the struts, and if you do a histology by a cross section, you see all the cross sections of the struts to the area, and you see that they're very weirdly distributed. So what we develop is a technique that is based in minimum cost pass approaches and some empirics. What it allows us to do is to actually simulate the insertion of a device. So if I look at this, this point, what we say is 20%, 25%, and 32% packing rate. Each color is a different coil. So you see about how many we had to put roughly. You tend to put between 6 and 20 of them, depending on the size. Remember, 1,000 pounds each of them, so it's quite a lot, depending on what you do. But what we observe is that with this algorithm of simulation, what we can do is insert the coil, say at 20%, then we take them out virtually. We add 20% again a second time. They will fall in slightly different positions, but it's still at 20%. And then we take them out. We put at a third time, so we have again in a slightly different position. So it's multiple. It's like treating the patient multiple times. And then I can look at the statistics of what happens in all of those. And I can do that at 20%, but I can do that at 25% and at 32%, etc. So if you do that, what you have is a plot like this, where depending on the packing density, you look at how much you have reduced the flow. And what you observe is that while at the beginning, the specific configuration has a big influence on the flow reduction, because exactly where the coils fall was important. As you increase the packing rate, of course, that doesn't matter anymore, because, and you go to a sort of asymptotic flow reduction value. And this is interesting because it means that there is a certain value beyond which it doesn't matter that you keep putting additional coils. The only thing you're doing is wasting money and potentially overpressurizing the aneurysm and rupturing it. But if you put less than this ideal number, then it does matter a lot the way you put them. And then your outcome might be very dependent on the operator and the exact way the coils were inserted. So this is exactly what was seen about 10 years before our study, which was for 2011-13. So this study is from 2002. And what you observe is that aneurysms that have a larger packing rate tend to be more stable compared to aneurysms that have a lower packing rate that tend to be unstable. However, the precise percentage of stability depends on whether the aneurysm is small or large and the size of the neck. So it's patient specific. Optimal packing rate and for a particular device as a function of whatever variable we want to make it as a function of. Finally, we talked about boundary conditions, and a lot of that is done through wearable sensors and machine learning on those. I don't have the time to go in detail over that, but we model. We have models that allows us to generate boundary conditions, which are age, sex, body mass index, match to the individual that we have. And we also can look at variability and uncertainty so that we can look at the influence of the stress levels or the influence of patient variability, for instance. Okay, so just to recap and conclude, so I talked a bit about ways in which you can build virtual populations, and I only scratched the surface. We are currently working in methods that use generative machine learning models to actually synthesize virtual populations. We talked a bit about virtual interventions, how you place stents and coils. You could also do the same things with models and simulators of imaging to look at virtual examinations. We talk about a bit about virtual outcomes, these flow reduction metrics, but you also could look at virtual readers through AI that do automatic measurements. And this is, a field where there is lots of work, connects to ideas of digital twins, virtual patients, as I mentioned before. FDA is already working as well on these, and MHRA, in terms of using these approaches to generate evidence. These papers written by FDA's colleagues, where they say, in silico imaging trials are cheaper, faster, better, safer, and more scalable. So that's a sort of direction of travel as well from regulators. And then I'm going to go to the final conclusions. So I hope I convey that there is a lot of progress being made in AI, but that most of the AI contributions that probably you have seen and I have seen are mostly around detection, classification, so really try to automate subjective tasks. That by this notion of computational precision medicine, adding modeling and simulation, we can expand on what conventionally AI can do, that allows us to integrate data from multiple modalities in these models, and also that fully automating all of these pipelines requires that we find ways to optimize parameters, automatically estimate the sensitivity, assess quality control of every steps, to be able to have fully automated and streamlined tools. And hopefully what I see is that in the future there will be an integration of AI with a lot of these more not only phenomenological but also mechanistic ways to model and bring in priors, either about the patients, about the diseases, or about the procedures themselves. And with that, I'd like just to do a quick thanks to the team, of course, in the lab, which are the people who have done a lot of the things that I've been presenting today. But also, remind you that we have the upcoming IPME deadline, and that if you haven't considered submitting something, you still have the chance to do so. And it's going to be next year in the beautiful city of Bariloche in Argentina, close to the Andes, and you're more than welcome to contribute and participate. And with that, a big thanks and open for questions.
Yeah, thank you, Professor Fernie, for the very comprehensive and actually insightful talk for introducing the concept of the in silico clinical trials on the virtual patients. Okay, so do you have any questions from the audience? If you have any questions, you feel free to unmute yourself to ask the question. I think we got actually one question from the chat box. So from Lee, what are the biggest challenges with the computer-aided diagnosis systems or methods in real-world hospitals today? So performance, easy to use, patient privacy policy. I think this is related to one part actually. So Professor Franji actually mentioned, yeah.
Yeah, so thanks for this question. I mean, this question I don't think has a single solution, a single answer, but I would say to me the biggest. So I'm also working with our local hospital in Leeds in deploying what I call AI 1.0 algorithms. So what I presented is mostly 2.0, if you wish. But the 1.0 version of things, the biggest problem is really that at least in the UK, the hospitals are not prepared to deploy AI. And what I mean by they're not prepared is, I don't think the problem is performance. I mean, of course, you can always do better algorithms. But if you compare with the performance that has today the clinics, I think AI can do a lot better already now. The problem is we are trying to develop, let's say, a FedEx concept without having airplanes and without having roads, right? So we are trying to develop AI algorithms and we are lacking the infrastructure where those algorithms will run. Hospitals don't have connected data. They don't have connected departments digitally within those. And they also don't have the infrastructures to deploy sophisticated computational pipelines. So I think the modernization of the infrastructures in the hospital is key. And current infrastructures haven't been developed for a data-driven era. They have been developed for essentially accounting and billing. And that needs to be rethink. And I think the hospitals that are more able to anticipate this transformation and move quickest to to have cloud-based infrastructures, the ability to have all of the data sets connected in an integrated packs with electronic healthcare systems, with the lab analytics, all of that will make the development of better data sets and consequently the development of better AI tools easier. Privacy, all of the other things, I'm not saying they're trivial, I'm not saying they're not important, but they are solvable.
Yeah.
With the technology we have.
Okay.
How
we also
far are asked?
Yeah, we
I'm
have
just
also
going
have
through the
additional
questions
here. Yeah, maybe Lu Yang can unmute yourself to actually ask your. Actually, you have two questions, right?
Yes. Thanks, Professor Frangi, for the very wonderful talk. I have basically two questions here. So the first question is about the selection bias part. So what would be a good training data for the generative methods? Say if the training data for the generative methods is already like biased, then would the generative model itself also capture sort of the bias inside the training data so that it will lead to another like biased or unfair situation for the downstream tasks? So that's my first question.
Yeah, no, it's a good point. Look, I think there are two ways to deal with that. One is by improving the data that you input in your generative models, and the other one is by having smarter algorithms that somehow can compensate for that. And I think both things are important. Because there is so much you can compensate algorithmically if your data set is too biased, right? So that's why one way to deal with this is moving away from small data sets. You really need to go to ideally real world data, which means, like, what I mean by real world data, Leeds region has a PAC systems that connect 3 million people. So if I have access to 3 million individuals, the ability that I will be too biased to one particular demographics is reduced, still is there, but is reduced. The other way is to look at population imaging studies like, China Biobank or UK Biobank, which are population studies. So they are not studies that are biased by a particular inclusion or exclusion criteria. Again, those still are biased, Like for instance, in the UK Biobank has 60,000 subjects, but is only about 10% response rate from the original people that were invited. So every 10 people that we invited to UK Biobank, only one say yes, which probably means these are the people who have better resources, that are closer to the places, that have money to actually take a day off and do all the examinations. So again, there is biases, but it's the best we can do. And then on the other side, there is the algorithms we can improve. Like for instance, could we develop ways that if we have, say, 5 or 6 data sets, that each of them has a bias, but a slightly different type of bias, could we look at ways to combine them into a single generative model that is less biased because has the information from all of those? And there is a slide that I skipped, which is the one about virtual chimeras. That is where we are trying to develop essentially that concept of blending different, is what is called in statistics, data sets of data sets, right? So how you bring those together.
Yeah. Okay.
Yeah. Thanks for the answer.
Yeah, I think the other question you had, which is about many other factors, the advantage is, again, if you look at real world data sets like the one I was describing, in 3 million people, the likelihood that you will have quite a few of them that have angiographic scan and have hypertension, or have angiographic scans and don't have hypertension, is much bigger than in a conventional trial with 100 patients, right? So the ability to work on real data will allow you to understand what are the different factors that affect the target population, and therefore understand their influence. And I think that's a bit what you're asking in that second question, is how we measure the efficacy under those different factors. Well, first of all, you need to be able to identify the factors that you want to check, and then you need to be able to model them. And a lot of those typical risk factors are modelable to a large degree.
Yeah.
I think
we also have
there's another
actually, thank you for the answer. We also actually have got another question, but I would like to rephrase a little bit, because actually this area, so with the medical imaging and also the computer assisted simulation, is a very dynamic and active area, and also it's a highly interdisciplinary. So also actually some because it's related to CS, medical imaging, that's the AI, machine learning, and also simulation, all these areas, and it's we really wanted to push these technologies into the clinical practice. I think maybe you can actually give us some suggestions for the undergraduate or postgraduate students who are actually about to actually enter this promising direction here. So yeah, so I think Mukai was asking essentially a question about this point, and I can see well, I don't know if it's a him or a her, but I can see the student clearly saying, this looks impressive, but how do I, poor computer scientist, get into all of this? And I would say, look, Mukai, I am also a poor electrical engineer, right? So all of us have a particular angle to it. And I think I would say, first and foremost, be very, very, very good in whatever you are. So if you're a computer scientist, be a top computer scientist in your area. And especially if you are at the PhD level, you cannot do a thesis with everything that I just was discussing. It's just impossible. I mean, what I summarize is work of 20 years, right? But the reason why I wanted to present this to you is to make you aware that while you need to have a strength in one domain, you need to be open to all of the other disciplines and understand that the big sort of advances in the next 20 years will be at the interface between disciplines. So learn to work with clinicians, learn to work with engineers, learn to work with people who are from industry, learn to work with people who are very different to you. So be good in what you are, but learn to develop, bridges and interactions with other communities, because while they will come to you for you being excellent in your area, you will not be able to make a big impact unless you work with people from other areas. That's kind of the short version of it that I can provide. I think there is another question. That's also actually my question. So I think maybe this is the last question, due to actually we already overrun a little bit later. So this is also actually my curiosity. I know actually you have been working in this field for many years, but how actually can you manage, so many disciplines here? As mentioned, this is highly interdisciplinary. So, yeah, can you answer this question? Oh,
For all of this knowledge, I think it was one of your colleagues saying, Kei Ikemura. I think, yes, you're absolutely right. And that's sometimes what I feel when I work with my own colleagues, that, that no matter how much time I spend in trying to understand their other disciplines, you sort of still miss always something. But that's one way of looking at this, is that the half empty glass, the half full glass is, it gives us a completely new perspective on our own domain. Like, for instance, a lot of the algorithms that we are developing are inspired by the challenges that are the challenges in computer science that are created at that interface. So what is very important is that you go out from your community to understand those interdisciplinary problems, and then you come back and you say, what can I contribute from my domain, right? Like, for instance, just to give one example, the idea that I think Lengwan was mentioning before about, Luyan was mentioning before about how you build these biases or how you compensate those biases. I didn't have the chance to explain that today, but there is a lot of work in core computer science going on in that space to solve and address that problem. But that problem would have never come if I just talked to people in the MICAI community, because they're all trying to solve segmentation and registration, and to some extent,
yeah.
I don't know you guys, but I get a bit bored of doing that. And at the same time, working with colleagues in computational mechanics, these colleagues are telling me all the problems of doing mesh generation. And then my question is, can we facilitate that by, other techniques that make computation of good meshes easier? So it's understanding the problems by talking to others, but then coming back and reflecting what is your contribution that you can make. And, as you get older, like perhaps I'm getting, there is the opportunity to work with the students and academic colleagues from other disciplines, and therefore build a very interdisciplinary team, which is the one we have at the minute. There's a question about, do you need to study medicine to work in your area? Not really. You need to, depends on what you mean by study medicine. If you mean doing a degree in medicine, I would say no. If you need to read things on medical journals and talk to clinicians, yes. And this is, I had the fortune that I did my PhD in the Netherlands in a radiology department, so I had a lot of exposure to that. But I would say try to get exposed as much as possible to understand clinicians and work with them. And sometimes they will drive you mad, the same way that clinicians may think that we drive them mad because we talk different languages. But, being able to speak across two disciplinary languages is very important. Okay.
Yes, I agree. Actually, we should have the ability to learn it, actually. So not actually, we don't need to master it, but we need to actually master the language, to communicate with doctors, interdisciplinary researchers. Yeah.
So, for instance, just a very practical suggestion. Sometimes, let's say, a student once was a bit unhappy because a clinical author was involved in his paper when he thought that the author, this clinician, hadn't made a lot of contributions. Now, the guy had provided all the data. But more importantly, what I said to that student is, look, if this doctor didn't contribute to your paper more, is because you didn't share the manuscript earlier with the person so that the person could actually give comments and critiques. So you left the manuscript to the very end, and now, there is very little that person can add to your work. So I would say it's very important that you involve clinicians in your publications, but do that early. Don't do that last minute. Don't do that as just because my supervisor tells that I need to add one. Do that by early on, by sharing your ideas, allow people to comment and criticize, expose your ideas in meetings and presentations to them, and allow yourself to be challenged and listen, basically. That's the main thing that will make you grow in this area, I think.
Yeah, thank you. So actually, we have already overrun like 8 minutes. So I have to end this session. So thank you everyone, and also especially actually Professor Franchi for the very comprehensive and actually insightful, inspiring actually talk here. So yeah, especially in the early of the morning. So if you have further questions, so we may have the follow-up communication or potential collaboration, because I do see actually some medical some colleagues from the medical companies or the other universities. Yeah, here. Okay, so thank you, Professor Franchi and everyone here. So this session is ended. Thank you.